what you have here, these two are specimens of the human brain. You can tell from the shape. What you have here, the smaller ones, and also you can see the shape, they are more oblong. This is more oval, the human brain. So these are your sheep brain, right? So you can see the dura mater, how tough it is, irrespective of the human or sheep brain. That's your dura mater. Okay, you can see that on this brain with and without dura mater. Remember that. This has the covering. This doesn't have the covering. Are you seeing or no? No. All right. So this pin is passing through the dura mater. Okay. The arachnoid matter is kind of difficult to see. So we have a picture for those of you who want. You can take a look. So you can see how flimsy the arachnoid matter is. Sometimes, depending on how you dissect, it goes on the back of the inferior side of the dura mater, or it stays on the pia mater unless you stick something in and lift. Then you can see it's very tricky. Okay. So we'll leave that alone. So what will be this layer? Assuming the arachnoid matter is not pia mater. Okay. So dura, arachnoid, and pia mater. What about this fissure? Longitudinal fissure. Okay. What about the lobes? Frontal. Parietal. Below that.
Temporal.
In the back. Occipital lobe. And those of you can see the cerebellum better. That's your cerebellum, right? So you can see the right and left hemisphere. So if it's pointing to the side, lateral hemisphere of the cerebellum. Vermis is in the middle between those two lines. You see that? That's your vermis that connects the two hemispheres. So you cannot miss those two lines. It's right in the middle. Okay, so here it's in the middle, and you'll see a bunch of worms. You're going to be having 1 hour to come closely and inspect. Okay, so don't panic. Let's move on. What is this C-shaped structure? Corpus callosum. Okay, so in the sheep brain you have to look carefully. It's also there, but you have to look carefully. You have to come closer and take a look when I'm done. Okay, and what is that space? Lateral ventricle, the membrane. Septum pellucidum. So this is who you are. What is the oval thing there? Thalamus. So that would be. You saw the pituitary gland in the picture. Now it's broken. You see, it's very delicate. So you don't see the pituitary gland here, but you can see on the inferior view. Can you see the bump with and without? That's your pituitary gland. It's been removed, so you can see the infundibulum, the passage. You see that? So your pituitary gland is basically like my hand on this needle. So assume my needle is your infundibulum. That's how it is. So when you break it off, it's gone. So all you see is the hole, the passage. Are you with me? Okay. So it's very difficult to see this piece that connects. So that's your infundibulum. Basically, this is infundibulum. They refer to the point of attachment, the passage. That's infundibulum. This is your mammillary body. See that round structure there? Again, that's your first bump, second bump. What is the first bump? Pons. The second? Medulla oblongata. The spinal cord starts there. And here again you see the lateral ventricle. There's no septum pellucidum. The ventricles, other ventricles, this is your third ventricle. It goes all the way around the thalamus, starting a little right from the lateral ventricle. It continues. So you have to think of them as connecting channels. Otherwise you are randomly figuring out where is what. So think of it as continuous channels. Lateral, third, aqueduct, cerebral aqueduct, fourth. Okay, then it goes out. All right. This is your pineal gland. Can you see here? You will see the thalamus. Below, behind that will be the pineal gland. It's a little behind and above. And again, you have to see this is my left piece. That's my right piece. So you have to orient yourself. The first thing you need to see whenever you see a specimen is to figure out where is my front, back, top, bottom. Then use the landmarks where things should be. Okay, otherwise you are looking at the right picture and you are looking at the left specimen, they are going to be a little bit off. And those are the 4 structures. Those of you who can see the colliculi, if you want to take a look, or you can do it after I'm done. Each one is a colliculus depending on where the pin is. And what else are we left with? That's your tree of life, arbor vitae. Okay, here you can see that also. Okay, so the only thing I want you to pay more attention, meaning, is this number 50 or whatever. In this case, this is your dura mater, but when it comes inside, we have to use the other name, falx cerebri. If it is there, it's dura mater. If it's here, falx cerebri. There, dura mater. There, dura mater. The same thing anywhere here, we call it falx cerebri. Okay. Did we miss anything? Probably not. So what I'm going to do, I'm going to